First up, we have Astrid Rademacher.
Okay, hi. So my name's Astrid. I'm from the University of Cape Town. I just finished my PhD. So this is actually the first time I'm using my official title in a talk. So thank you. That helps my existential dread a little. So I just wanted to give a talk basically about how I've used these very kind of commonly used packages in my PhD work and how it's really elevated the kind of analysis I was able to do. Okay, so I work on a resurrection plant called Xerophyta schlichterei. It was called Xerophyta viscosa. Well, we thought we were working with Xerophyta viscosa. And then halfway through my PhD, we realized, oh no, this is schlichterei, and now I have to say schlichterei, which is not very exciting. But basically, resurrection plants are capable of desiccation tolerance. And what this means is that they can dry to equilibrium with the air. So think of like a piece of biltong, and once you add water to it, it resurrects into a cow. Not quite, but the same kind of principle where we have a totally desiccated cell, totally desiccated plant, and once you water it, it comes back to life within a few hours. So I just want to give you an idea of what this process looks like. So this is Xerophyta humilis. Just added water at the beginning, and you can see it's starting to regreen already. Within 3 days, it starts putting out flowers. Look at that, so sexy. Okay, and this is Miraculously verbellifolius. It's actually more of a shrub. And as you can see, after 8 hours, it's starting to unfurl and resurrect. I'm enjoying all the wows from the audience. Thank you so much. And this is Craterostigma pumilum. It's another resurrection plant species. One of the great joys of my life was going to Buffelskloof Nature Reserve, where we get our plants, where we collect them, and actually observing these plants occupying very similar niches altogether. So you'll see like the greatest hits of all of these plants like in the same niche. It's so beautiful. So my project is actually on cellular death. So senescence is a process that happens. We're all doing it right now. We're all dying. But basically it's what happens to older cells. It's a very natural process, but it can also happen in response to abiotic or biotic stress. So, salt stress, heat stress, stress, from pathogen infection, that kind of thing. And basically what happens is plants completely sort of kill themselves and redistribute their nutrients and it kind of proceeds from older tissues towards younger tissues. So the older tissues are kind of saying to the younger tissues, go on without me. And to reproductive organs so that the plant can quickly reproduce before it dies. So this is a very kind of genetically hardwired process that happens in all desiccation sensitive plants. But how are resurrection plants, which are desiccation tolerant, kind of shutting off this supposedly hardwired process? Cool. So this is Xerophyta schlichterei, it's a cutie. And here you can see it in the fully hydrated state, looking all cute and bushy. By the air dry state, that you would assume that that's dead. If you saw this in your house, you'd be like, RIP, I left you, I neglected you, bye. But within 24 hours after rehydration, it starts to look pretty good again, right? And then after 48 hours, it returns to the same state as the full toga. So it's a monocot resurrection plant, and so it's our model for understanding how we might improve drought tolerance in cereals, which are also monocotyledonous. So we know a little bit about, well, actually quite a lot about how it's able to survive the desiccation process, but we don't really know how it represses the genes that are supposedly sort of hardwired to be responsive to stress and signal death. Cool, so the research questions I wanted to answer are when during drying are these senescence processes initiated? So in Xerophyta schlichterei, every time it dries down, only the apex of the leaf will actually succumb to the drying. So only the very, very, very tips of the leaves will actually die, and the rest of the leaf will go into a metabolic sort of dormancy inactivity, which it means it's still viable. So how does this process actually happen in these very little tiny apex of the leaves? And how is it regulated? So what is actually happening on the genetic level to regulate this process? And kind of the question that I've opened up a lot of avenues to explore but haven't really tackled head on, which is kind of the most important question, is how is a non-senescent tissue actually preventing the cellular death process? So we have a really nice model where we have enough tissue in a plant to to compare the non-senescent and senescent tissue during desiccation. This is the first time this kind of work has ever been done for a resurrection plant, which sounds really cool, but when you're doing it as a PhD, the first time you like actually piloting a project, it's not pretty. It's quite very unpretty. Anyway, so I just want to give a quick overview of what genes actually are, because I realize that not everybody knows this. So they're little pieces of DNA, not little pieces of DNA, vast pieces of DNA that live within the nucleus, and they encode something called transcripts, which are kind of messengers that are copied off of this piece of DNA, and that's what happens when we have gene expression, is we actually have the gene being copied into a little message. And finally, we have transcripts that are translated into proteins, which are the sort of end product of this gene expression process. And so we can actually sequence genes and transcripts and proteins, and we can quantify them, and we can compare them between samples. So this is what I did in my study, was I compared the transcript level expression of genes. And these proteins are, there's many different classes of proteins, but I just kind of broadly outlined a few here. So enzymes, so those are the proteins that actually catalyze chemical reactions with inside cells. So things that are responsible for whether you can eat ice cream or not. So if you, if you have an enzyme that's defective, you maybe can't digest lactose, and that makes you run to the bathroom, for example. We also have signaling molecules, signaling proteins. So these actually communicate within cells and between organs of plants and animals and all organisms. And then we have regulators as well. So I want you to remember that little regulator part. So these can actually influence this transcription process, the translation process, and they also regulate how maybe enzymes work. So the regulators that I want to talk about in this talk are actually regulators that control this expression of genes. Okay. So people often ask me, like, how do you get meaningful information out of a plant? Like, I don't think people understand like what it takes to really get sort of gene quantification or transcript quantification from plants. So I just wanted to give a little outline of how we do this. So we torture our plants by drying them. We withhold water, and we sample for relative water content. So we measure the water content of the plant as it's drying down, and we store tissue away. And when we've decided that we are enjoying a relative water content range, or we think that that relative water content range might be interesting to actually look at, then we'll dissect tissue from that sampling point. And this was me in the beginning of my PhD, grinding samples in liquid nitrogen. Very scary at first, was totally overwhelming. We extract them in this compound called trisol, which once you centrifuge it, phase separates your sample into RNA, which is the transcripts that we're interested in, the DNA, which is the genome level information, and then everything else, which is the proteins and lipids and cell debris. So you can actually take this RNA, put it into a very fancy machine that I don't understand, and it'll give you information about how those genes are actually expressed. And so we can compare that between hydration states and between tissues. So in this study, I compared the senescent tissue, so the apex of leaves, to this kind of pre-senescent zones, this transition zone, and then the non-senescent tissue, which is viable after rehydration. Okay, so I just want to show you guys quickly what a drying curve looks like, so what the leaves actually look like. So this is the apex of the leaf, probably about the last, so how many centimeters is that? 7 centimeters of the leaf. And so you can see by, when you rehydrate the leaf, you just don't see it coming back. And we can also see this in the relative water content. So this is the hydration status of the leaves. For the non-senescent tissue, it's a sigmoidal sort of curve down. This is an air dry state. When we rehydrate it, it gets water very quickly into the tissue, whereas the senescent tissue doesn't actually recover its water content. So something is happening in here somewhere, one of these sort of water content ranges, that's causing the senescence process to occur. Okay, this is, I think, a big slide, so it's taking its time. Cool. Cool, so once I got my actual gene counts, you normalize those gene counts, so that's the transcript abundance. You normalize that to the size of the gene and to the sequencing run. And then you get this data called TPM. And basically you can plot that as a time course, so that's what I did here for each tissue type, so non-senescent, pre-senescent, and senescent. And then I ran k-means clustering on it. You can do this with a bioconductor package called tmixclust. It's quite nice. And so I got these 12 clusters that describe my data quite well. And so it was kind of like disconcerting at first. I thought, oh my gosh, there are no differences here between my senescent tissue and my non-senescent tissue. What am I doing? And then I realized that the main differences are actually happening really, really late in the drying cycle. So around 10% water content is where you see the spike in gene expression in the senescent tissue. So it's waiting until really late in the game of this drying cycle to actually switch on these genes that encode death, potentially. So that was really cool. So we wanted to dig into that a little bit deeper. So usually when people compare conditions with gene expression data, you will compare it to like the hydrated state, right? Because I had 3 different tissue types, that didn't really make sense for me, because if I compare my air-dry senescent tissue to my full turgor state, I'll be getting both genes associated with drying and genes associated with death, and how do I tease those apart? So I compared them to the non-senescent air-dry state, and dplyr was like my friend here, man. Tidyverse, shout out. Just for I don't know if people here work on transcriptomics, but consolidating annotations from multiple sources like BLAST and Mercator and that sort of thing. It's just so nice to just bring those all together with Tidyverse. So I really recommend. Cool. So this is my senesome. I was told recently that it's kind of like not really cool to call every little minor data set of genetic expression information an ohm, but it's an ohm. Don't care what the haters say. Cool. So I just basically displayed this data as a heat map. So where it's red means that it's more significantly expressed in whatever condition compared to the full turgor state. And then I made this last column here, which is just the senescent tissue compared to the non-senescent tissue in the air-dry state. So you can kind of see how the gene expression is changing over time for all 3 tissues, but you can also compare the air-dry state to one another. And this was kind of like the greatest hits of death. So protein degradation, DNA cleavage, it's just, it's tickets for these poor cells, shame. Also signaling hormones that are to do with cellular death and transport of nutrients. So we were pretty convinced, okay, this data that we actually dissected out from those senescent tissues at air dry was actually the data that signals for death. I just want to quickly show you guys my app that I made. So I turned it into an app because ain't nobody got time to print 30 extra pages of their thesis. So I made it into an app for the planet, guys, for the planet. So basically it's just displaying these heat maps, and you can download the data if you're interested. And also you can search for genes that you might be curious about. And it also gives us a little indication of how the genes are changing in each tissue type. And also some clustering information. So from the k-means clustering that I did, you can also see where those genes are actually coming up. And then I also organized it into different tabs based on the actual cellular process. Oh, God. Oh, sorry. This is, okay, I'm going to repeat myself. Sorry, guys. Haha! Look how cute it is. Cool. So I was just saying that you can interact with it. Thank you for that little hand at the back. So you can actually interact with it. I don't know if I can actually, yes, I can. Okay, scroll. Yes. So you can scroll down and search for your gene of interest. You can download the data, and then you can flick between tabs to see how the genes are behaving in different sort of categories within the plant's repertoire of gene expression. Cool. So I also made one for the actual for the desiccation related genes. So if anyone is interested in how plants are able to survive in the desiccated state, I'll hook you up with that as well. Cool. Next. Oh. Buffering. Okay, cool. So now we know which genes are actually responsible for cellular death. And they're only expressed in the most extreme conditions, but how are they regulated? So how do we dig into that regulation? Cool. So I just want to go back and say that I lied to you about genes, because they have a promoter region and a coding region. So this coding region is the part of the gene that actually is copied into the transcript. And the promoter region is where those transcription factor binds. You remember those regulators I was talking about? Those bind to this promoter region, and they encourage binding of this RNA polymerase, which is actually what makes your transcript. Okay, so we can actually look at the sequences of DNA over here in the upstream region of the coding region and predict which transcription factors might be binding there. And so I wanted to find out, are these transcription factors under control of the same promoters? And so are they being sort of upregulated together in concert? Cool, so I made this giant co-expression matrix for all of my genes, so comparing gene 1 to gene 2, and only connecting them if they're correlated over a certain threshold. So the threshold that I chose was 0.94. And this is what the network actually ended up looking like. So here I've mapped my k-means clusters to the network as well. And so we found that there was this nice little, very discretely clustered subnetwork that was the senescence genes. So I wanted to see, okay, are these genes being controlled by the same transcription factors, the same regulators? Okay. And then I used this really nice piece of software. It's not R, but I really advocate for it. It's in the browser. It's called Dreamy, I would say Dreamy, of the Mimi suite. And basically what it does is looks at all the sequences in your upstream regions of interest and compares them to a control group. So in this control, I compared it to my whole network to see which DNA sequences are actually enriched. And I found that there were these two sequences that were actually enriched, and there was a G sequence and a C sequence, or just a G sequence. And you can see the whole subnetwork is controlled potentially by the same transcription factors because they have the same sites in their promoter regions. Okay, and then I actually went and looked at, okay, what transcription factors are actually in the network. And there's this really nice tool as well called TomTom, where you can compare your transcription factors of interest and your binding site to a known database, and it'll give you ones that are known to bind in plants, which is really useful. So we were able to identify the transcription factors, or rather predict the transcription factors that are probably responsible for the senescence phenomenon, which is really cool because from this we can this is kind of the launching off pad for a bunch of projects that can look into how this, this process of senescence is not occurring in the non-senescent tissue. So maybe what is binding to regulators or preventing their action in the non-senescent tissue. And this is super cool because if we can get crop plants to do the same thing, that would be really, really useful because the first step in creating something that's desiccation tolerant is getting it to not die. Logic. Yeah, so how are the non-senescent tissue blocking this response to prevent cellular death? And yes, can we use this to actually prevent cellular death in crops? And I just want to say thank you guys so much for your attention, and thank you to my wonderful group who's supported me a lot, and to the R Consortium for funding this trip, and shout out to Heather and Emma for all the admin help. Thank you so much. You can find me on GitHub. I have some code on GitHub, but it's not very good, so please don't judge me. I'm getting there. And my little shiny app and ResearchGate, and find me on Twitter as DataChino. I also just want to give a quick shout out to Exegetic. I did their course last year, and that really helped me so much with actually digging meaning out of my data. So if there's a course that they're running in your neighborhood, definitely go. Cool. Thanks so much.